Oh, just I forgot to on my that audio because maybe those who are in online just waiting for your voice. That's become certain limitations in hybrid mode. Concentrating at one, so another people may suffer. Okay, so that is like ATCC and MTCC. Just I was talking, right? Microbial type culture collection center and an ATCC American type culture collection. There are so many other also. Australia, other Japan, and mainly the China. Leading guy in all the categories. Rather, you can say the America now. The Chinese people has moved very high. Whether it's bacterial research, fungal, or any human related event. Currently, in a patent status and the IPR and everywhere, they are far away from the entire world. So they do have their culture acquisition center. When you are looking to buy or purchase, there is a treaty between the two countries are there. They will may give you or may not. You are ready to pay any amount, but they may not provide. That's also depends. This is the basic culture or something else. Okay, so there are some authorities are there behind it. So this was the one point. Okay, second points. Types of a bacterial culture medium is there. Another is like a nutrients agar. That is like a simple peptone water. So here sometimes it may be written in two ways. Maybe it is solid or liquid. Both can be possible. It is like agar, that is a complex, blood agar, synthetic, Davis Mingoli are there. So what are basically parts are there? Blood agar. Same thing is distributed like simple and the complex or the synthetic or the another terminologies are also coming the mixed. That is a natural source and synthetics are being mixed together to prepare certain kind of a particular kind of a media. So why people are using or preparing it? The kind of a bacteria, those who are working on it, it's required to get isolated. So it's depend upon the bacterial suspension or the bacteria we are looking to isolate from any of the plant. So nutritional base, another is a functional. Here we need to think more into it. So it's a basic media. That is like a compositions being unknown, right? That is a nutrient broth. Nutrient broth having the major thing is what? Generally, that is having the yeast or the beef extract. Further, it is a sodium chloride. Okay, these are the 2 to 3 only are aspects are. Then one another is a peptone. Peptone is a like protein digest. Maybe it is from the animal or the plant. It's starting from the source is like peptone, then the salt is the NaCl, and another is like a yeast extract or beef extract, third component. So it having all the possibilities into it. That is like a carbohydrate, lipid, everything is there. Whole suspension of it, it's dried and mixed. Fine. So it is a nutrient bath, maybe in liquid. Another part is in a solid medium. When you are going to add the solidifying agents, particularly the agar, and the starting it may happen. Then the selective media. Now our select part will start to work it out. So there may be, we may add certain inhibitory components. Okay, like if you're going to add the certain kind of antibiotics, that means maybe gram negative and positive. I hope maybe you are knowing the difference between gram negative and positive based on the difference in their cell wall, right? We are classifying them in gram negative and positive. So certain are very specific antibiotics are inhibitory for the gram negative. Certain are for the gram positive. So if you are looking to isolate, I am looking to get the gram positive bacteria out of the soil suspension. What do I have? So in that conditions, I may go for it. Why? I'm looking to only work on the gram positive and negative. What could be the reason into it? Can you think? Why the selective media are required, or what could be the requirement of it? Why should I go for the sometimes the gram negative or the gram positive bacteria on? When I'm looking to isolate something, I should open for the all. Okay. But I may become like a selective sometime, right? Or your mentor has told that we are going to do only a gram positive. What could be the reason for such kind of a selection? It is like analysis. I think you are going to have the such questions also, right? In your examination. So that is why I'm opening your mind in that way also. Just a teaching is not a required. It should be the opening of your mind is also, it's only come after the understanding. So understanding and analysis based, Why we need to go for the selective media? I think you are telling something.
Because it needs some, specific characteristics. The reason why we are choosing it, we need some specific characteristics, not all of it.
Yeah, right. Special characteristics. That means when you are working, suppose I want to get some antibiotics out of it. We know that my culture, it's a it is having the certain pathogens and I want to control it. And I'm going to use the another weapon from the other bacteria. So if it is the gram positive, I should use the gram positive or the gram negative. That is the one question. I don't know about it. I want to control one. So I'm just trying to look upon their activity. So I will classify like a pathogens is the one kind of a group A is there, and I want to control with them. So certain antibiotic producing bacteria I'm going to isolate. So I may start with either is a gram negative or the gram positive. So I will go for the selective method. I will isolate all the gram negative separately, gram positive separately, and I'm going to mix with them. Okay, with my pathogens. I'm going to find out antagonistic effect. Maybe you're knowing antagonistic effect means one is going to inhibit the growth of the another. Sometimes may happen that like you may have learned like symbiotic association, asymbiotic associations, and so many others are there. But among them, it's like what are the antagonistic effects are there that one will not allow the growth of another. In that conditions, we are looking for the certain kind of a special inhibitory agents. So it will going to only allow first gram negative and positive, and this gram positive I'm going to mix with my population of the pathogens. It is the basic part people are working and doing while doing the research. Currently, we know it's like a mucormycosis is persisting at a very high level and growth. It is because of fungi. It is a not job of only just bacteria, one, to control the fungi. It is like a very giant. Itself, the fungi is releasing so many kind of inhibitory agents, and it are not going to able to control by the bacteria. Okay, then we may require the bacterial consortium. Bacterial consortium means it is having the mixture of the bacteria. We don't know it about one, but it is on from the certain side, and we have collected, or maybe we are knowing that one bacteria is producing some kind of antibiotic, another bacteria is having the certain antibiotics, but some inhibitory chemicals are being synthesized. We may club together all and try to control one fungi. So what you can do, you may develop the fungi culture on your plates, and you are going to mix it. Either it is in liquid or semi-liquid, or you are working in the liquid ones. Okay, kind of experimentations we may design, and you are going to add such kind of bacterial populations, and you are looking that my fungal growth is inhibited or not. Then we need to go for the selective medium either, or the first selective medium application is started, then we apply to another place. So many variants are coming while the designing any sort of experimentations. So in each part, we need to understand that what could be the use and applications of any kind of media. I think yesterday when we went in the lab, right, there you have seen that LB broths. Some different broths names were there. 4 to 5 broths were there. Peptone broth and other all are available. So why we have just started with nutrient broth? Because it's a basic one. We are not particularly working on the specific. So that is why we have started in that way. Okay, so as per our applications and our desired agenda, we need to go for the different aspects. Fine. So it is a selective medium. Yeah. Yeah. It's happened. Bacterial consortium, when you are preparing, that is why I have started with gram negative and positive. It is like that you are in a class, right? If someone is coming from the another green t-shirt guys are coming and sitting, you will start to look, right? But sometimes maybe it's a new guys, same t-shirt, colors, everything, you will notice, but not that much. The difference is in your hand. Okay, maybe you have thought that some new guys is there, new fellow may came, admissions or something is there, but you will notice that someone is a new, but you are not going to directly react. Okay, but it is a green t-shirt, Okay, something is there. Why is he is sitting here? Okay, so it will be get noticed. It is like gram negative and positive. That is why we are separated. While preparing the consortium, they are having some unity. Yeah, among them itself, gram negative, they are going to fight with each other. So while preparing the consortium, we need to understand their basic characters. They may fight with each other. Sometimes our motto is what? To control the fungi. Even though they are fighting, suppose we have made a collections of 21 isolates. Out of 21 isolate, 5 to 6 are having antagonistic effect within amongst self. 5 or 6 get died, 15 may remain. Okay, that's not a problem. Let's think that this 15 able to control this fungi or not. Because in shortest time research we are looking, then certain points we have to neglect it. But when the time is there, you are working about a 6-month dissertation is there. First week you have completed. Now you may go for addition of 2 to 3. You are going to look upon the antagonistic effect is there or not. So you may able to go for the permutation and combination. Maybe you are knowing that maybe you have learned in your past permutation and combinations. That is a isolate number 1. I'm going to mix with isolate number 2. Isolate number 1. I'm going to mix with isolate number 3. Okay, it is like a combinations I'm preparing. Okay, while preparing the combinations, is there any antagonist effect is coming? Then they are going to control one to another. There you have to understand the bacterial colonic characters. That one is inhibited another's growth or not. So it is very complex. It will take about not only a 6 months. It may go for about a year. But when the time is limited, don't worry about what they are fighting with each other. It is like one project is given to whole class, right? When it is one group, that is some different aspect. Okay, so I have given the 5 groups. Maybe among your 5 groups, I have given task, and who is going to coming out very well, it will be get some rewarded. So you may start to think about that, what they are doing, what we are doing, how we can make it a better. Okay, you are keeping a secret mission like that, but you will looking to find out what the another is doing, right? But if it is given the some another way, you can work it out in different aspects or having the same aspects, 2 phenomena. Different aspect is there, just the terminology is given the environmental control. Then you are having the so many things in your head, and you can have the so many projects. One may go for the sewage treatment plant, some is looking for the industrial effluent, some may think about the air pollution, some is looking for the noise pollutions. Okay, four groups are there, they have distributed. But if I have given the one only, the industrial effluents treatment related to it, using your microbial applications, now the competitions will be a tougher. So it is my desire to start and design the experimentations. Accordingly, the output will come. But the time limitations is very high. Then I may give in a group, sometimes in a on whole class. Whatever is going to coming out with some students are coming, I want to answer for the problem. Okay, so problem solving can be done in various ways. Good.
Certain bacteria after like inhibition, certain they like they are mixed with the fungi. So they are showing less activity. So how you can increase that activity?
Yeah, that may be. It is like a 6 organism is fighting each other. 15 has able to control it. At the certain level, it has done. I never mind. But government is asking for the some conclusive medicines. So I have done fastly. Now the second stage will come. I will going to superfine. So I may go with the combinations. Then I'm telling that 21 I have isolate were there. I may go with the 5-5 group in combination first, and I'm going to check. Then I'm going to add the 5 plus 10. It maybe happens. So it is the time taking research. It is kind of a research, and when you are doing certain kind of research, it will take time. Just not experimentation. Okay. So when it is Corona has came, they have started to give the some kind of specific drugs. Maybe this or that, and it has worked. Then the people has accepted. If not worked, people will say that this drug is not working. It was a totally ab initio. Okay, ab initio, that means we are not knowing right now. So we are forced to do certain kind of a work. Okay, at that time, whatever the result will come, positive and negative, that you are going to note it down and report it. because nobody is knowing in the entire world about it. It is a totally new word. Then you are going to super scrutinize it. That now how can I improve it? So combinations may be given. Like some medicines were given in the first time. Then the variants has came for the corona. So people have started to give another drugs given. Then the another third combination has came. So amount of it is been reduced and increased. So it is totally experimental and the research. So here it is particularly I'm talking about the mainly applications, and I'm going to relate with many aspects. Maybe it is agriculture or the pharma. I'm mainly concentrating relate to certain in pharma. It became easier for you to recall or the understand. Okay. Certain selective bacteria like a thiosulfate citrate, biosalt sucrose, TCBS, which is selective for the Vibrio cholerae. Some few examples are there that you may able to recall or remember. Okay, sometimes it may be asked into like objective questions in a way that's straightforward. What is the usage of TCBS? So you're able to recall like a Vibrio cholerae. Fine. Another is like McConkies. Okay, so few are there. It is like that from the childhood we know that a sun rises in so and so directions. Okay, like that we have to recall and remember. In same case, we have to recall and remember few at least example. Okay, currently you are not going to experimentally all you are going to do, but at least for the as a B.Sc microbiology students, they able to remember usage of certain kind of a medias. Okay, another is like McConkies for the enterobacteriaceae. Differential media, yes, it's used for the one type of form to another. It is like a difference in the colonic characteristics. What's going to happen? Example, menthol salt, eosin, McConkie. Yeah, here one by one we are going to look. Menthol salt agar. Differential media for the fermentations. The color of may change. These are the very particular bacterial media have been used. That if you are looking to identify or differentiate, or whether you have the same isolate or not, that maybe some kind of bacteria is there. Color agent is there, like black color, red color, something is there. When the bacterial suspension start to grow over it, you surprise them next day, the color of the whole plate may change. It is like from the green to yellow or yellow to purple, or so many colors are there. So what generally what is there into it? Some specific dye is been added. Particular dye is been added, it going to react with their extra. That is like a bacteria are generally secreting certain kind of a chemicals out of it. So they are going to react with it. Like enzymes you are learning, right? Enzymatic assays are you are learning. So bacteria are generally do two different things. Is there the dye is taken out of it. Dye is there outside in their environment. They have engulfed it. It is been processed. So during the process, it may be like that some compound may release, and because of that, the color of our media may change. So we may going to particularly identify whether the selective component is there or not, that we know to confirm. So confirmatory test is also there. Like if you are having the E. coli, or we know that it's having the certain capacity to convert this color to into another. So then we are going to check it out, whether we do have the E. coli culture or something else is there. B. mega is given, or B. bacillus is given, something is there. So for there, we may go for certain kind of a mediums. Another is like a eosin, which is differential for the lactose and sucrose fermentations. What are the basic part is there? That when you are looking the sucrose or lactose, we know that lactobacillus we may able to get from our buttermilk. Right, buttermilk is there when the lactose fermenting bacteria. So you want to study some basic components related to the buttermilk. So over there, what you can do, you may collect, or you are looking to that which bacterial suspension or the which strain is very much efficient in doing certain fermentative process. So what you can do, you may purchase buttermilk from Amul, Gopal, any other so many companies are coming, or you may take the isolate from your indigenous at home. You came from it, you have collected into one scientific tube. Many times here, I don't know how many experimentations we are going to perform in this semester. I was used to give the certain activities related to the milk. That the milk is how much having like pasteurized milk we are calling, right? And another is like a raw milk is there. And a third maybe a category that is a boiled. That is you have boiled it at your home. So what's going to happen that normal flora of an animals that is from the either is the cow or the buffalo. There, when the churning of the milk is done, it is going to transfer from their mammary glands to our milk. If this raw milk, if you have started to eat as a part of food or you have drink, okay, what's going to happen? There is a two chances that a normal flora from the cow that has came our digestive system is able to destroy it. But certain time in disease condition, there may be two disease conditions are coming in the animals. One is related to their animals itself, either in their body or sometimes on their skin part. Mammary gland is required to be cleaned first before taking a milk. But by chance, it is not performed on the skin. One kind of bacterial populations may be there, and another is the cow maybe get infected. So normal flora were disturbed by the certain pathogens. During the churning of the milk, it was been mixed. Now my milk do have normal flora from the cow plus two kind of a bacterial populations. Now it has been came into my food as a raw material I have started to eat. There may be now two chances. That pathogens may harm me, may not, or maybe get destroyed by our system. Okay, that may be maybe it's next semester. Immunology, have you learned? Immunology, one of the subject is coming in this year, not now. Immune system. Okay, so that is there are two kind of innate and acquired immunities are there. Don't worry about it. So we are not going to dwell upon it. So there is now two chance. That cow milk I have taken, it may going to harm me by chance sometimes. Certain pathogens, it is in a when we are mammals. Cow is a mammal, human is also a mammal. Certain pathogens are sharing that capacity. Okay, then I may get feel ill. Not ill, but sometimes a diarrheatic condition. That pathogens are not going to harm directly. Okay, but they may disturb my normal flora in a gut. There is a two possibilities are there. Some students, green color, some guys have came. If only one fellow, you are not going to get any problem. But at least 15 or 20 guys are coming. You are only at about 21. They have started to come and the 15 students has came. You will not going to notice, you are going to manage them after. You don't know what is their purpose, why they have been called to be here. Okay, and by chance it may happen that you are going to have some interactions, maybe a positive or the negative, right? So in that case may happen that they may disturb my midgut normal flora, or if they are highly pathogenic in nature, they may start to give fever and other conditions. Okay, so what I need to do, what I need to understand over this in experimentations, that raw milk is having the how much bacterial population. Okay, so another is the pasteurization is enough, having the capacity to kill this normal flora as well as the pathogens or not. Third is the boiling. So what we can do, like a methylene blue as the several other kind of a, you can say dyes are coming. Generally, basically, we may go for the crystal violet or the methylene blue. You may take it out the 10 ml. So generally, I'm giving the scientific tube to many students in a group, in a labs we are performing. So some people, what they can do during the morning time, some people are telling that at our home, some the people are coming, and they are giving a milk to us. Some are telling that we are using the Tetra Pak. Some are telling that we are using the so-and-so pouches from somewhere. So what just I'm telling that to get that milk in the is possible 2 in 3 forms. One is the raw. Okay. Some people are coming to give to at home. That is the raw milk. Amul pouch is there raw, but it is pasteurized. We know already. Third, it is like that raw milk, you have boiled it at your home, then you have taken in 10 ml. Now we are coming in a lab. Okay. What we can do, we can have some set of protocols with us. We may keep the certain tubes as a control sometime. Or this from the same tube, you may split it out. One is kept as a control. And in another, you may going to add some kind of a, you can say, a dye. Dye is been added. We know that the color capacity. Blue color is there. Light blue color is coming. Incubate it for 24 hours. control as well as this. Otherwise, you may keep the control in refrigerator. So we are going to retard or the hold the growth of bacteria. So milk can be stored for 24 hours. In incubator, you have kept same milk. Okay. And some amount of you have added the dye. Next, you are coming, you are going to match or going to observe set tubes. So you will surprise that the color intensity maybe they had used in some tube. In some tube, it is as such. So what could be the possibilities over there? If just question. If color intensities remain as such, what you can conclude? And the color intensity has reduced, then what you can conclude? Can you say, anyone? Same. First. Okay. You have tried to, but you have confused yourself. You have mixed both the answer. If fermentation may happen, something is there, then the color may intensity reduce. If no, okay. Anybody else? Changed or decreased, intensity is decreased. Yeah. Yeah. Very nice. Fine. Very good.
If it has not changed, that means the pathogen have but is something else that we change.
Very nice. Very nice. Yeah. Yes. The question was there itself. That is also you need to segment. I have given the 3-4 option. Raw material, one was the pasteurized and the boiled. So you have given the answer only for something and something. Okay, you are all near to the right to the answer. Now I'm going to ask one by one, or you may think about it one by one. That is a pasteurized milk is there. Okay, and there is when I have kept for the incubations for 24 hours. Next day when I'm coming, there is a no color intensity check. If it is such, then what you can conclude? Okay. Huh? Yes. So it is pathogen free, bacteria free. So no color change. Okay. Second one was of boiled. Okay, that milk was boiled. There also there is a no color change. That means this now became a fully free of pathogens. Now the question is only coming for the raw material, raw milk. Two students have, or the two groups have came with the two different research. One having the as such color, second is the decrease in their intensity. So what could be the conclusive points? There's a no color change. Maybe that means there is a normal flora, maybe from the animals, that is from the cow, are not able to digest it either, or there is a less amount of bacteria is there. Okay, bacterial population is going to be there, but maybe a less, and it's not maybe that much harmful for us, and we can take as a raw material. But another raw material, raw bacteria, okay, they have reduced the intensity. That means it having the, that may do have a population of the bacteria rather required amount, and they have digested our dye, and it is converted into something else, into some other compound. The intensity has reduced. So there is now two possibilities. That raw milk may have the normal from the animals, or maybe a harmful pathogens also are there. So we can test only with the two and next stage. Okay, maybe You are taking the simple, just do the experimentations. You are taking the raw milk. If you get ill, that means some pathogens may be there. If it is not there, we can just basically identify. But sometimes some people are taking daily raw material, they are not going to get a problematic. So it depends upon the immunity. But we are looking to identify, then we need to go for the isolation of the bacterial populations from there. So such experimentation is designed accordingly. So where that time we need to go for selective media, because there will be a normal flora, maybe a lots. We want to selectively find out a lactose utilizing bacteria or the sucrose based or certain particular, then we need to go for the selective media itself. I hope now the point is clear. So that is like a milk-based experimentation is there. I'm not so sure right now. Maybe it's in microbial physiology. This experimentation is there or not. If you are comfortable, time will be there. We'll do certain extra experimental parts. You will like it in a way that what you are doing daily, right? It's we are going to perform in a lab. So you will be surprised that, oh my God, that a milk I'm taking, it is really, to be said, is a good, but it is not. Like Tetra Packs, you can buy from market, and the same in accordingly this. What could be the output? So you can identify. Wait, so how much time is there? Oh, it is about. Okay, it's about to 85. So I'm stopping here. We will further continue in next lecture. Attendance link will be there itself. You can just put it down.